Welcome everybody to the inaugural Ruth Lynden-Bell Lecture. This is a very exciting moment in the history of our college. First, I'm going to just tell you a little bit about Ruth Lynden-Bell. Sadly, she isn't well enough to be with us, but we have just admitted her as an honorary fellow virtually in another room. So Professor Ruth Lynden-Bell is a chemist who has made crucial advancements in the knowledge of the properties of liquids and disordered solids with abnormal structures. You can tell that I wrote all this myself, can't you? And she has pioneered new techniques over her career, identifying previously unknown key physical processes using simulation and theoretical development. She's an emeritus professor both at Cambridge and at Queen's University, Belfast. And as well as being an honorary fellow, as she became today, she's a fellow emerita here, and she was acting president of the college. And she was and is an extraordinary woman, and I would be grateful if you could put your hands together for her. And now I'm very pleased to welcome another extraordinary woman who we're very fortunate to have with us. And I'm not going to give a long talk about you because you're going to tell us about yourself, and also I might not be so convincing. But I know that Susan Galbraith, as well as having this fascinating title, Executive Vice President, Oncology R&D, AstraZeneca, is one of the most important people in the world in terms of developing drugs against cancer. And actually, I know that to be true because I told one of my friends who worked for Roche, the enemy, that you were coming here. And I said, oh, we've got this person coming from AstraZeneca. And she said, oh yeah, who is it? And I said, well, she's called Susan Galbraith. And she said, Susan Galbraith? You've got Susan Galbraith coming. Wow, that is fantastic. And then I discovered from various other people that I had thought that you were a really important person, but I had no idea at all just how important. So thank you very much indeed. And now I'm going to hand over to Susan. And after Susan has spoken, Kristin-Anne Rutter, who is Executive Director of Cambridge University Health Partners, will chair a panel with Susan and our excellent and esteemed further guests. But I'm not going to introduce you in detail now because it might be too much for people to take in. So, Susan, over to you.
Oh, thank you, Dorothy, for that extraordinary introduction. I'm not sure I can live up to it, but I will do my best to give you a little taste of how I've got to be where I am today. And what I want to do is something that's a little bit different from some of these discussions. So I want to give you the slightly raw, honest review of life to this point, as opposed to the polished, lovely external view that, well, people often get, because I have a reaction to the sort of social media world that I think, frankly, contributes to mental health challenges, where everybody assumes that it's only them that struggles and everybody else has everything down. It's not true. It isn't true. Not true for me. It's not true for anybody else that I know. So let me give you a little taste of how I got here. And I cannot but start in this esteemed institution, but acknowledge the fundamental importance of education in enabling people to live up to the opportunities that they would have. And the reason why I show you the picture on the left-hand side is my grandmother worked in the cotton factories in Lancashire. She had little choice about her career, little choice about any aspect of her life. And so I don't take for granted the fact that I've had choices and opportunities that were not granted to my grandmother. The picture in the middle is a picture of my mother. My mother wasn't able to do science at A-level because she was a girl. Wasn't an option that was available to her. So like that, again, just one generation back, range of opportunities I've had because of the access to education is fundamentally different from the previous generations, not to be taken for granted. So to the privilege to be here and to be able to have the kind of discourse that we can have in these kinds of institutions is simply not to be taken for granted. I picture her as a graduate because she graduated from the University of Manchester. She did an economics degree. She did that part-time whilst working as a teacher on her own at 19 because she didn't really get on with her stepmother, who had to leave home at 19. So some of the grit and determination that I think is an enormous part of life has come from my mother. A picture of my father, who passed away earlier this year, is there because my father was a physics and maths teacher. He also grew up in the centre of Manchester in a place that he described as the slums of Manchester. And he was determined that our life was going to be fundamentally different than his. All three of his children ended up at Cambridge University, one this and the other. So you could say that he was remarkably successful in that goal. And again, it was through education. Having fought for the empire in Malaya, he came back and then went through and got teacher training and was at one of those schools that he met my mum. So that's a little bit of the background, and I think we're all shaped by the backgrounds that we have come from and the experiences that we've had so far, but that has to be a real part of the story, and I just want to acknowledge them. So, as I've already shared with you, my family come from the north of the UK. I started off at Manchester Medical School, the Oxford Road campus at Manchester Medical School. When I was at Manchester, I met the guy that I'm still married to now. I'll tell you how many years ago that is. One of the most important things you can do in life is choose the right life partner. I was blessed in that regard and have had an awful lot of support, as you'll see, through the years. But one of the things that happened is I did an intercalated degree after my first 2 years at Manchester. He was in the year above, had graduated from doing engineering and then started to work for Ford Motor Company in Essex in their R&D facility. So I spent a year commuting between Manchester and the south. And I didn't particularly enjoy that third year. So then I applied to every single medical school in London to see if I could transfer and do my clinical training in London. I was rejected from every single medical school in London. I think there were about 16 or 17 then. A lot of them were consolidated. So there was a fair amount of rejection that happened during that point. Now, some of them rejected me with a single line. I won't say who. The Royal Free, which obviously has a history of encouraging women in medicine, wrote me a page and a half about how they'd love to have me, but unfortunately the University of London's rules wouldn't recognise the exams that I'd done at the University of Manchester, so I'd have to retake my first 2 years. And whilst I love my husband, that was a little bit too far. So it said at the bottom of this letter, have you thought of applying to Cambridge? And I thought, well, no, frankly, I had assumed that they would be less keen on having me than the University of London might be. But I thought, well, I've been rejected 16, 17 times. One more time really isn't going to hurt my ego at this point. And as it happened, I fitted into the Cambridge system because I had this intercalated degree in the Bachelor of Science in Physiology. And so I fitted into the system, and the Cambridge Medical School at that point was trying to take on more of their graduates. So a happy coincidence. And so I did my clinical training at Cambridge Clinical School at Adam Brooks, and I'm an alumnus of Robinson College. So that's kind of how I started. And I tell you the story because it gives you an idea of grabbing opportunity, not being worried about rejection, and not being worried about failure. Failure is often something that people have a lot of fear about. Actually, It's a great opportunity for learning and taking on to the next thing. It's a fundamental part of science. So we should actually embrace failure as an opportunity to learn. So one of the things I then did, having graduated in medicine, I didn't get the SHO rotation that I wanted. I thought I wanted to be a cardiologist. I ended up doing a rotation with oncology, and the rest, as they say, is history, because I fell in love with oncology in Cambridge. I then chose to do a PhD, and I'll be completely honest again here, raw and unplugged, that one of the reasons that I chose to do a PhD was because it enabled me to have a period of time when I could guarantee that I could work during certain predictable hours and not have to do on call. And I applied for my PhD having carefully selected the institution on the basis that it had a creche that was available, which was at Mount Vernon Hospital. I was at the time I applied for my PhD 7 months pregnant with our first child, and I delivered our second child towards the end of the PhD. Now, none of this would have been possible without a lot of support from a range of other people. Again, this is a nice picture of my husband, who on a previous talk I forgot to mention. Right? Did I mention that learning from mistakes is a really important thing? So anyway, the pretty picture in the middle. So those are our two children, Jennifer and Alex, who now both work in Cambridge. One in a biotech, the other's training to be a patent attorney. I guess so. They've kind of worked out okay, I think. In the middle picture, the things that I did during my PhD, I worked on a drug that affected the microtubule structure in cells, and these green patterns that you see here is a microtubule cytoskeleton in a cell that helps the cell to keep its shape. These are endothelial cells, or the cells that line your blood vessels. The red part is the actin, which is a different kind of cytoskeleton element. I spent a lot of time looking at the cell shape in my PhD and looking at these green things. It would all be done automatically today. I could do the entire 3 years in about half an hour. That's okay. I learned how to do that. And then the picture on the right is a dynamic MRI image that looks at tumour blood flow. So this drug that I was working on for my PhD worked by changing and disrupting the microtubules, changing the shape of endothelial cells and making the tumour blood vessels more leaky, which helped to shut them down, and that affected, and we looked at that with dynamic MRI. I'll come back to the physics that my father taught me was also going to be fundamental. He was right, because I had to relearn all of the physics when I was doing my PhD. So that's kind of how I started. And then, again, serendipity happened. So at the end of my PhD, the drug that I've been working on that did all of this was licensed to a large pharmaceutical company in the United States from the small biotech that owned it. And that pharmaceutical company was called Bristol Myers Squibb. This is their campus located outside of Princeton. And so my husband gave up his job, and we moved with our two small children across the Atlantic, and I became the sole breadwinner, none of which was on the original plan, I'll just point out. Several things happened here. 2 weeks after I finally arrived, this is having planned all my life carefully, they killed the drug that I'd worked on for 5 years. And my illusion about teaching this massive organisation about the mechanism that I was involved in disappeared. And so I was bored for the first time in my life, which was actually quite a scary experience. So I wandered down the corridors to the Discovery Labs and talked to some of the people in the Discovery Labs. And I met a woman there called Mireille Jurikanko, who lent me her copy of Reut's Immunology. Now, when I was at medical school, another couple of mistakes that I made was I finished 2 courses and I thought, thank God, I can throw the textbook away. I never have to look at this again. One of them was immunology and the other one was statistics, and I was wrong on both counts. Okay, so I had to relearn stuff. I had to relearn immunology. And one of the things I did while I was doing that was go to a small conference on immuno-oncology, because in those days they were very small because nobody believed that the immune system could be targeted effectively with drugs to increase the benefit that you get with treatment and help overcome the progress of cancer. So I went to this small conference and I saw some of the data that was presented from a small biotech with a very exciting compound, which is now called ipilimumab, which is the first of the immuno-oncology drugs that made it to market initially for melanoma. And I was involved in the licensing of that drug. So again, had they not killed the drug that I'd worked on for 5 years, had I not had a clean slate and had to go out and learn, I would not have had the opportunity to be involved in what is a really big immuno-oncology revolution in the treatment of cancer. So that's another good lesson about it. The other thing is about burning bridges. Had I been able to turn around and go back to what I'd done before, I probably would have done in those early weeks at Bristol Myers Squibb. But I was the sole breadwinner. My husband had given up his job. We had 2 small kids. I had no choice. I had to make it work. And there's a really good lesson in the careers about that, which is the level of commitment that you need to make and the fact that you actually see through periods that look a bit dark and gloomy, and you never know what is going to come through on the other side. This is a picture of my daughter, slightly older now. This is, in the Chesapeake Bay. It was one of the things that we got to do, and I just love it because it's a sense of adventure, the excitement that you see when you see something new, and that was one part of what was my experience of 9 years at Bristol Myers Squibb. And then I came back to the UK for a job at AstraZeneca, which I knew was going to be a turnaround job. I'd grown up through the Bristol Myers Squibb organisation. I was heading now the early oncology and immunology team and doing the biomarker development. And I was asked whether I'd be interested in coming back. There was only probably one job that I would have come back to the UK for, and this was it. So I knew that we had to sort of take that opportunity. One of the things that happened very early after I came back, so we moved our family across the Atlantic back into sort of a nice place in Manchester. And then within a couple of years, there was a change in CEO and a strategic plan to move to Cambridge. And it was like life's going round in a circle again. I went down to Cambridge. So this is some of the team that was around when we were obviously having a team meeting in Cambridge. And again, I didn't know what I was letting myself in for at the time that I came back, but it's been a very exciting journey. Lots of ups and downs, lots of difficult moments, but lots of incredibly exciting moments as well. And now this is my broader team. I'm now responsible for all of Oncology R&D, right from the very start when we start to think about what kind of targets that we want through the discovery, the early development, the late phase development. I have a team that's global, goes across China R&D, Japan R&D, and this is this fabulous group of people. And it's a privilege to be able to work with such a diverse group and background. So when I reflect on my experiences of my career to date, what am I most proud of? I've had the privilege to be involved in the development of 12 new medicines that are now available for patients with cancer. And that was one of the things when I was a practicing oncologist, I felt really strongly that there weren't enough tools in the toolbox to offer to patients. There weren't enough things that you could do. And the thing that I'm most proud of is when I hear the stories about patients who have benefited from one or other of the drugs that I've had some small part to play, because it's a massive team that's involved in the development of any drug, some small part to play in those medicines, and you hear those stories. We share a couple of examples. A young woman, I say young woman, younger than I am today, so therefore young, mid-40s with 2 young children, who was diagnosed with multiple brain metastases from her lung cancer. Never smoker. Some cancers that you get in the lung have a particular mutation. They tend to be more common in non-smokers. Actually, probably diesel particulates may have a role to play in the development. But what we managed to do is see a benefit for her with one of the drugs where she had a complete response in her brain, and she lived for long enough so that her children could remember her because they were small at the time that she was diagnosed. Those kinds of stories are the motivation that keep me going when there are difficult days, and there are often difficult days in what we're doing. So that's one part of what I'm proud of. The other part is just an illustration of the fact that over the years that I've been in this role, and these roles at both Bristol Myers Squibb and AstraZeneca, I've had the incredible privilege to work with a large number of very, very talented people. And this is just a few pictures of some of the key people that I've been involved in. And so when you see people develop and develop their capabilities and go off often into other roles or other companies and help also make the field better through that, I get an incredibly warm, fuzzy feeling from that. Also collaborating with other organisations, the American Association of Cancer Research UK, Academy of Medical Sciences, the connections that you can build and that network is really important. Often, in life, the most important things are not the things themselves, but the connections between things. And this is one example of the fact that this network of connections has been an enormously rewarding part of the career that I've had today. So my advice to any of you in terms of your careers, look, life, truthfully, in its unvarnished form, is full of challenges, difficulties, opportunities, highs and lows. So you've just got to make sure that you have the energy and the support around you to support you through both the highs and the lows. I think that's really important. Packing a mirror. I can tell you as part of two large organisations, I've probably had more psychological tests than have been created on the face of the earth. I've had an awful lot of feedback. That feedback is a gift. You have to be ready to receive it, okay? So you've got to be ready for some of the reflection, because again, what you're trying to do is learn as much as possible along the way. Learn as much as possible so that you can be even more effective in terms of having the impact in the direction that you want to do. And to do that involves a bit of self-reflection, and not everything that you'll hear is as glowingly positive as the very kind introduction that Dorothy has given me. I've had other feedback as well that is less good. I'm apparently a bit impatient, for example, and people do point that out on occasion. I've benefited also from a huge amount of advice from other people, and I'm deeply grateful for people's willingness to help. And that was the other piece of advice I would give you. You'd be surprised how willing people are to help you if you only are willing to ask for the help that you need. So I've benefited from a number of mentors. Annalisa Jenkins, unknown to Dorothy, was one of my early mentors at Bristol Myers Squibb, always gave me perspective. Elliot Siegel was the head of the R&D organisation there. Two guys in the middle, Di Chaplin on the left and Gordon Ruster, were my two supervisors at my PhD. And then, I have to mention Gordon Mills, who has been the chair of my scientific advisory board, a group that I set up when I was at AstraZeneca that helps advise me. And my former boss, Jose Basalga, who very sadly died of Creutzfeldt-Jakob disease, just over 3 years ago, which is deeply sad. I'd known him for many, many years, and I follow many aspects of his advice on a daily basis. So what I would say is, look, we are social creatures as human beings. Build and use your network. Often people think that if you just put your head down and get the work done, everybody will notice. Wrong. They won't. And you'll benefit so much more by learning from other people through the network that you've got. So build it and pay attention to it and feed it, because you will benefit from it and they will benefit from it, and we'll all benefit from it if you do that. And the second piece of advice is, look, career is a journey, not a destination. At every point along that point, you've got to be overall enjoying what you're doing. And making sure also that there's a little bit of time for fun, because I'm a strong believer in fun. This is one picture from a bit of fun sailing a boat on the Chesapeake while I was out there. It takes time and effort. You've got to self-preserve. You've also got to persevere. There will be tough times. Rely on other people in the team, but have fun along the way. So that's a little bit of background about me. Now I want to tell you a little bit about what we're doing at AstraZeneca Oncology R&D and how I see the future of cancer treatment evolving. So first of all, I view that we're in a golden age of cancer drug discovery, and it's come off the benefit of technology. I think it was Sidney Brenner basically said that the technology evolution enables the discoveries that we've got. So you can see here on this chart some of the discoveries of some of the science that then enabled the application of that science into medicines that have subsequently come through. And this is just a chart of the FDA oncology approvals per year. And what you can generally see is, look, there was a long period where it was relatively static, but it has and is making a difference, and it's accelerating. And I think it's come off the basic understanding of the drivers of cancer and what is needed to improve it, and also improvements in the technology about how we can use that to address that. So that's very promising. What difference has it made in terms of the outcome? So some cancers have really very good outcomes. Okay, so the diagnosis of itself, although it is feared, and of course in the news today, a lot of information in the news about the fear that's associated with the diagnosis. I think the goal really of what we're trying to do is remove that and have better understanding, because from knowledge and understanding comes the opportunity for new development. So you can see at the top, we've done well with melanoma. Melanoma, because it's on your skin, typically you can diagnose early if people pay attention to it. And for the ones that are diagnosed very early, there's a very good outcome. Breast cancer also, because we have screening, and because also we've got treatments, endocrine treatments as well as chemotherapy and radiotherapy have improved the outcomes over the years. And you've got some of the hematologic malignancies, non-Hodgkin's lymphoma, chronic myeloid leukemia that have also improved dramatically. CMLs, because of the discovery of that BCR-ABL gene, the development of a drug that was able to address that specific genomic aberration. Lung cancer at the bottom, work in progress. A lot more that we can do here, and a lot more that we should do here, but much less progress than we've made in the others. So, why is that? Well, part of the reason is that in contrast with what I said about melanoma that you can spot easily because it's on your skin, and because of breast cancer that you can screen for, we haven't routinely used screening tools for lung cancer. So the majority of patients with lung cancer present with advanced disease, and this is just the data for Canada, Denmark, Sweden, UK, and you can see on the right that depending on the stage that you're diagnosed at, the outcome is very different. So, people are a bit nihilistic about lung cancer, and they're nihilistic for two reasons. First of all, there's a stigma associated with the diagnosis of lung cancer. Quite often, the first question that patients who tell other friends or colleagues that they've had a diagnosis of lung cancer say, "Oh, did you smoke?" Just think about that. Did you smoke? I.e., did you cause this yourself? And even if you smoked, are you less deserving of compassion for the diagnosis of cancer? Probably because you got addicted to it a long time ago. But many cancers that are in the lung are not associated with smoking. Radon gas, diesel particulates, there are other causes. If you have lungs, you can get lung cancer. And if you're diagnosed early, we can do a better job. So we should diagnose you earlier, right? And the screening tests are now available, and they're going to be targeted at the patients with the highest risk, but I think that can be broadened. So here are some of the current treatment options for lung cancer. Surgery. If you've diagnosed early, an option for surgery and surgical resection. External beam radiotherapy can also be given, and that can be given with curative intent. Chemotherapy regimens. And then more recently, targeted therapies, and as I've just talked about, some of the immunotherapies are now being shown to be active in lung cancer. So the range of options, tools in the toolbox are increasing. But the outcome, if you're diagnosed when it's spread outside of the lung, is still poor, as I showed you on the previous graph. So what can we do to improve these options? Well, the first thing is that chemotherapy, which is a standard of care for many different cancers, is actually given as combination chemotherapies, 2 or 3 different drugs, typically in combination. And those drugs were developed over the last 40, 50, 60 years, initially just based on the fact that they killed cells that were dividing quickly. And of course, not only the cells that are in the cancer are dividing quickly, but the cells in your skin that affect your hair, the cells in the lining of your gut that affect, diarrhea and nausea, they also get affected. And of course, the bone marrow, those cells proliferate all the time. And so what that means is your white blood cells go down, you're more susceptible to infection, etc. And that's one of the key risks. So can we do better? Well, I think we can. There's a new class of agents that are called antibody-drug conjugates, or ADCs. And what they do is they take the technology innovation of an antibody, which can target very specifically one of the proteins on the outside of a cell, and you can see that illustrated on this graph on the right. So the protein on the outside of the cell is illustrated here. Oops, can get this to work. Here, these things. This is the antibody that binds very specifically to that, And then the chemotherapy is attached to the antibody. So instead of the chemotherapy going everywhere, the chemotherapy only gets delivered to the cancer cell that has one specific protein on its cell surface. Key here is that you need to make sure that you're targeting something that is expressed on the cancer cell and not as much on the normal tissue, or as little as possible on the normal tissue. And so we've got maps of different proteins that are expressed on different cancer cells versus what they're expressed on the normal tissue. So that's one thing that can improve it. We've been putting antibody-drug conjugates into clinical trials for over 20 years, and it's only recently that we've really had great success in those making a difference, because the design of this system has had to be optimized. So the initial design, they put very, very toxic molecules on. They call it, everything has to be weaponized in cancer. I don't quite know why we use the terminology of war for treating cancer, but these are called warheads. Right, bear with me. They're just the cell-killing part. They put very, very toxic on, and what happens is in the peripheral circulation, a little bit of that can drop off the antibody. It's not completely stable. And so that ends up like just being given something that's very toxic, generally systemically. And that was the first reason why these drugs didn't work very well. You remember, failure, opportunity to learn. Okay, so then people modified them and put slightly less toxic things on. The next thing is that they tried to make the linker very stable so that they didn't fall apart in the peripheral circulation. And that worked a bit better, and there were some drugs that were there. But the problem with that is when they're very stable, they don't then get broken down inside the tumour cell, and they don't get released. And so you're not killing all of the tumour cells within the cancer. The cancers are always, it's a bit like when you treat with a less than fully adequate antibiotic to a bacterial infection. If there are a few bits that can get away, you generate resistance really, really quickly. So that wasn't great. What we now have are linkers that are stable in the peripheral circulation, but cleavable in the tumour microenvironment. And that can also, once they're cleaved, can go across from one cell in the microenvironment to the next. So this cell might not have as many of these receptors on the surface, but it's still going to get killed because it's quite close to this one. So the evolution of the technology around the design of this system has taken 20 years, and now we've got some really, really good antibody-drug conjugates that are coming through, and more that will come through as well. So I fundamentally believe we can improve on the standard of care chemotherapy for many, many different cancer types, and many biotechs and many pharmaceutical companies are investing in doing this across different tumour types. So that's a great promise. So we can improve on that chemotherapy backbone. The second thing is about the immune system, which I talked about briefly. So cancers typically take 20 or 30 years to evolve. And your immune systems are very active in that process. So the reason why more of us don't die of cancer is because your immune system is actually very good at spotting abnormal cells and killing them off. But gradually, over time, you've got a co-evolution of the tumour that's got this evolutionary drive and the immune system that's trying to prevent it come through. So you get this balance. Your immune system is designed to recognise self, so it doesn't destroy normal tissues that you've got in your body. It's designed to attack when there's a viral infection or a bacterial infection, because that's the evolutionary drive for that, or abnormal cells. And then in some situations, the immune system is also designed to not attack, to be quiescent, to defend. And so pregnancy, for example, when you've got, a new organism with a different set of genes and a different set of proteins, you don't want that to be rejected by the immune system. So then you get into an immune privileged state. And similarly, when you're healing after some of these attacks, that's a relatively immune privileged state. So what a cancer does is hack into these systems and make it abnormal. And what we want to do with the immune system is recruit and engage immune cells that can recognize that it's not self and stop ignoring it, that go into the attack mode and come out of this defend mode. And we can have mechanisms that address each of those. So I think the first immunotherapies have worked and are making a real difference, but we can do even better. And the technology to understand what we can do and how we can engage the immune system is ever improving. And there's hope. So here you see some data from a trial It's not an AstraZeneca trial, so I'm showing other people's data here, with an immunotherapy and chemotherapy together. And you can see here what these are called Kaplan-Meier curves. So you start off with everybody in your sample is well and surviving, and then if somebody has a progression event, you drop off down this curve over time. And you can see that in the subgroup of patients that have this particular protein on the surface that tells them that the immune system is in the kind of exhausted state, and you give this drug that switches it out of that, you can have a really good outcome. But if you don't have that, you have low levels of it, the immune agent doesn't work as well. What's also really interesting here is that if you can get, and these are patients that are treated in what's called the neoadjuvant setting, so before you've gone into surgery, you can treat the cancer, and it's the tumour's there, the immune system and the cells are inside it. That's the best time to activate the immune system against it in that neoadjuvant. You can cause some shrinkage of the cancer during that treatment, and then you go in and have surgery, and the surgery is more likely to be successful, and that drives the outcome. The other technology that's come along that's helping us in the treatment of cancer is something called circulating tumour DNA. So it turns out that the cancer cells that are around your body give off a little bit of their DNA into the bloodstream. You can pick it up with very sensitive tests these days with sequencing. And you can, after the people have had this treatment and had the surgery, if you haven't achieved what's called a pathologic complete response, which is the PCR here, so pathologic complete response means that when the pathologist looks down the microscope at the sample that the surgeon took out, they don't see any cancer cells, right? In that situation, patients do really, really well. Look out here, 3 years, 4 years, much better than that 5-year survival. But these patients who still got a little bit of abnormal tumour-based DNA in their bloodstream, they do much worse. These are the patients that we can target for new treatments and make a difference and try and bring them up to this line over here. And again, this theme of circulating tumour DNA means that even before the surgery, you can identify patients that are at higher risk than that. So the tools and the technology to work out how we treat cancer are going to be fundamentally important to evolving how we care. So what does that mean for our approach and our strategy? So the history of cancer, and I recommend a book called The Emperor of All Maladies by Siddhartha Mukherjee, if you want to get some background reading on this. The history of cancer is the treatment of combinations. The advances in childhood leukemia, for example, have been through combination chemotherapy regimens. And the future of cancer treatment is going to be through combinations. What we've done in AstraZeneca is build a portfolio where this is what we call the colour wheel. On the right-hand side of this, you've got different mechanisms that attack cancer in a different way. So you've got the antibody-drug conjugates I've talked about, and radioconjugates, where you can stick a radioactive isotope on and still direct it to the cancer. We've got agents that affect the repair of DNA damage, which is a fundamentally important mechanism in cancer. You've got mechanisms that target specific mutations in specific tumor types, and then you've got agents that target what's called epigenetics, which is the overall control of which parts of your genome get read and not. And then the left-hand side is all the ways in which we're activating the immune system to better treat cancer. So you've got classic immuno-oncology, the agents that I've talked about that move cancer out of an exhausted state, the immune system out of an exhausted state into an attacking state. You've got immune cell engagers that pull new immune cells into the cancer for those cancers that don't have enough immune cells there. And then the latest revolution that's happening is cellular therapy, which you can think of as living drugs, where we take cells from a patient, take them out, genetically engineer them to recognize the cancer cell more specifically, and put them back into a patient. They home in the cancer and kill it. So we've got a portfolio of drugs that can attack cancer in many different ways, and we are planning to put them together. That's one part of our strategy. Second bit is where we treat in the course of the disease. We can use these circulating tumour DNA tests now to diagnose cancer earlier. Actually, in the UK, there's a collaboration going on between the UK government and a company called Grail to use a multi-cancer early detection test using this circulating tumour DNA technology. And I hope that will mean we can identify diseases like lung cancer and pancreatic cancer at a much earlier stage and give better options for patients. As I've already shown you, I think we can monitor during the treatment and switch treatment using this kind of technology. We're also, of course, using data science and AI across all of the things. We're using it in our discovery programs. We're also using it in our clinical trials. We're using it to inform how we design those, and with better models. I think this is making a huge difference to the speed of drug development. We're also better able to diagnose cancer and understand which of the proteins that each cancer is expressing on its surface using something called computational pathology. So rather than just a pathologist looking down the microscope, identifying the cancer cells and working out whether or not they express high levels of a particular protein, you can do it automatically with computational pathology. And finally, we're able to surround a patient with better care. So these antibody drug conjugates that I've talked about often have side effects, right? They're not side effect free, just like chemotherapy has. One of the side effects can be inflammation in the lung. With a digital app, where people just take the pulse oximeter and their pulse rate and the things, we can diagnose the onset of that side effect at a much earlier point, intervene better, and avoid people getting severe side effects. Digital health is happening. The pandemic actually accelerated this hugely. But you put all of this together, and that's how we make a difference with cancer. So how I see the future, you've got this expanded toolbox. You put it together with better diagnosis, diagnosing earlier. You put it together with new ways of attacking it to build actually combination regimens that can be transformative. You surround people with better care and better options for the patient care. And with that, and also by doing this and having some of the clinical trials done remotely, we should be able to include more diverse patient populations into our clinical trials. Very important point, very important piece for us to do. And with that, I hope that what we can do is change their survival curves and get them closer to the flat. We're already making a difference doing that. I'm going to stop there and take any questions. Thank you.
Well, thank you so much, Susan. We're going to save some questions for the end, if that's okay. My name's Dr Kristin-Anne Rutter. I'm an alumni of this college and also of the Cambridge Clinical Medical School. And so it's a real pleasure to be here to honour Ruth Lynden-Bell and to hear Susan speak. I was struck at the beginning by your comment about the power of education and the step back in time, 1 or 2 generations.
Yeah.
And actually I was reminded I was in Nigeria last week, actually, of all places, where I met 400 women who still don't have those opportunities. And so that also linked to your point of equity and diversity of thinking about how what we're doing here in Cambridge really does need to adapt out for the rest of the world as well. But after leaving clinical school here, I had a mixed career. I was in management consultancy, I worked as a doctor, I worked in the NHS, I worked for a startup company, I worked for pharma, and I am now back leading an organisation called Cambridge University Health Partners, which brings together the 2 universities and the hospitals to try and make sure that this fabulous ecosystem continues to be a place where new life science breakthrough discoveries are made, as we have in our history, where those technologies can be rapidly demonstrated and trialled and tested using our populations that we have here, and then we deliver very strong healthcare, both to our local population, but also spread that out to have global impact to improve health. And so in that role, that's all about bringing people together. I was very struck by your comments about network and working with people, etc. And that's what I spend a lot of my time doing, of how do we make the most of different parts of this ecosystem. So it's very appropriate this evening to be able to invite up Dr Bristi, Basu, and Kevin from Bicycle, who, if you come up on stage now, we'll give them a little round of applause. Thank you. And they are coming at this question of developing cancer from slightly different perspectives, all probably lots that you can relate to in Susan's talk, but one from a biotech perspective and one from an academic and clinical and healthcare perspective. I'm going to steal Dorothy's trick and let them say a little bit more and introduce themselves briefly, and then we're going to have a little conversation about those different environments, how they all work together, how they work with Susan at AstraZeneca, and then we'll open it up to a more general discussion. So, Bristi, maybe tell me a little bit about yourself, what you're doing now,
Okay.
and where you work and what you work on.
All right. So I work at the University of Cambridge, and I'm an academic medical oncologist. That means I spend my time in oncology. I'm a practicing doctor, so I see patients. I came from the clinic today, and I spend a lot of my time in clinical research. So I do have a research background. I have done laboratory research, so I did a kind of an integrated degree, a bit like Susan, and then really enjoyed getting my hands into the kind of lab work. And so I did do a PhD on a target which is very interesting, and I'm not going to talk about that today. But I realised that it was teaching me a lot of key skills and a lot of transferable skills. But actually, I really enjoyed the medicine. I was trained as a medic at Oxford, and then went around. Trained a little bit in Australia as well, and Cambridge. And when I came back from Australia, I really thought, oh, well, I'm going to be working next to the beach. So I thought Torbay, Cornwall, something like that. And so there was an interview. Yes, there's an interview coming up. Yeah. Exactly, there's an interview coming up. I'll do a practice interview with Cambridge because I'll never get it. And then I got it, and I was like, oh my God, this is one of those sliding doors moments. And it's like, well, what's going to happen? And I guess like Susan, it's sort of like, you make your decisions and you stick with it, you commit to it. You just say, right, this is how my life is changing, but I'm okay with it and I'm going to see it through. And actually, it was a great move coming to Cambridge. It's been a brilliant place, very nurturing for kind of female academics, I believe. And I think that through my work, I kind of went into the lab, did a bit of drug discovery, realised I wanted to go back into patients, and so did some work in the Marsden in drug development, and then came back as a consultant in early phase trials. So that's a phase one, the earliest sort of trials in human patients, where based on kind of laboratory data. And that's what I've been doing pretty much since then. So now I co-lead the Experimental Cancer Medicine Centre here at Cambridge, and my specific focus is in cancers which are still at that very challenging stage, which Susan has mentioned before. So when I remember when I started as a trainee in oncology, lung cancer, we only had one or two drugs, and they were all, it was a pretty dire situation. And now the number of options they have is phenomenal. I'm kind of treating some cancers which are still in that that stage, which hasn't got very many options. So I deal with tumours of the pancreas, the liver, and the bile ducts. And what's very exciting is that even in the short time that I've been a more senior consultant, I've seen a sea change in some of the management of, based on some of the technologies and the kind of genomic precision medicine approaches, the immunotherapy approaches. I've seen a sea change in at least a couple of these tumours. The pancreas is still one, which is I'm going to call a challenge. We're just waiting for our moment to come, basically. But now I interact with companies, with academics. We have a big experimental cancer medicine centre network UK-wide, because we try and build our academic partnerships. And then we interact with our pharma and biotech partners to try and get some drugs to patients. And yeah, that's what I try and do now. So I kind of try and do
the rough split of your week between clinical trials.
I kind of do quite a lot of clinical, I find, which is actually fine. Which is fine, And it's one of these questions is why would you want to do that? And I love that immediacy of that contact with the patient. And I just want to remember one of my patients now, who's sadly just passed away last week, but was a very important member of our patient group, a patient and public representation group. And he sent us videos of his daughter's wedding, that he managed to go to because we managed to get him well enough and those sorts of it's more than an anecdote. You feel like you're invested, and I feel like that close contact. I'll never have the same impact on cancer mortality as someone working and developing 11 drugs and seeing them to market, but I'll see the immediacy of the patient in front of me and their stories and the hope and everything that comes with it, which is a real privilege.
Okay. And a different environment, but probably lots of things you can relate to, Kevin. Do you
I
want
have
say
to
a little
say,
bit?
I wish I'd had that advice 30
Which
years
one?
ago
Which bit?
from Susan.
Yes.
I mean, that was just
failure or the
amazing,
network?
amazing. Just the
Yes.
how to approach your career was amazing. So, yes, very, very inspiring. So I'm Kevin Lee. I'm currently the CEO at Bicycle Therapeutics. This is we're now a publicly listed on the Nasdaq in the US biotech company. We are mainly focused in oncology. The technology originally arose from Sir Greg Winter. Many of you will know Sir Greg. And he looked at an antibody which contains about 1,500 amino acids. And he said, you know what, there's only 20 of those that actually do anything that's meaningful, interact with the antigen. So we took away all those other amino acids and we're left with these bicyclic peptides, 2 loops that are covalently linked to a small molecule scaffold. The idea being that the small scaffold replaces all of the amino acids in the antibody. And these molecules have a number of features which I think are really very exciting from a perspective of precision medicines. So we have 3 molecules currently in clinical trials. All of them are doing very well, very happy with the progress. And I think we've come a long way since I joined the company. When I joined the company, we were a startup. We're the first Cambridge, UK company ever to float on the Nasdaq. We've got 3 molecules in the clinic and a whole host of other molecules coming through. So very pleased with the progress we've made with the company. Where I came from, I've always been fascinated with research. I decided I was going to be a pharmaceutical researcher when I was 13. So that's a bit boring because that's all I ever
How
thought
did you know
about.
that existed when you were 13?
I have no idea. But that's where it all started. I've never developed 12 drugs though. That's pretty remarkable. Developing one is very, very tough. To get 12 is just unimaginable actually. So my career, very traditional pharma type research scientist, GSK for a number of years. Then I went to Pfizer in Boston. My family hated the US and wanted to come back as quickly as possible. So I was telling Susan, I commuted for 18 months to Boston. It's a very long commute. Not Boston in Lincolnshire, Boston in Massachusetts. And then one day I was approached about this amazing technology, the bicycle technology. And that was a moment. I had 4 or 5 offers of jobs coming back to the UK. And I just fell in love. There was one slide, I still remember the slide, and it's like, this is going to work and it's going to blow everything away. Haven't quite blown everything away yet, but we're trying hard. So I committed then, and this was a company that many people had written off, had been struggling for 4 or 5 years, and we slowly built it up, and now we've over 300 people. We have sites in the US, sites in Cambridge, in the UK, and 3 active programs. So.
And what is different of working in your small biotech now compared to those days of Pfizer and GSK? Or
I say to my colleagues who are in pharma, when you come to biotech, it's like living. It's like living. It's not, sometimes everything's a little bit soporific in pharma. Depends on what you're doing in there. If you're a research scientist, whereas you have major highs and major lows, it's a little bit amplified. I'm sure it's the same for Susan, but these are like one molecule in your pipeline, and you have a problem with it. That's like you're facing the end. And then when it goes well and you get the results, it's just, remarkable. So. And we were discussing just before, actually, it turned out that you're trialling one of his...
we're in an academic trial. So, the sorts of clinical trials, my research is in these early phase trials, and so we do academic collaborations either in networks or with Cancer Research UK, or we work with our biotech or pharma partners to deliver commercially, sponsored studies as well. And so, yeah, we're talking about a shared kind of
We're setting up new
shared
collaborations.
project.
Yeah.
Exactly, it's always networking. Exactly, but it is incredible that you meet people coming round, and how often people who are interested, you see them in different guises, and maybe even different companies, or a lot of academics tend to do. It is a lot more flexible now. I think the structure of careers is much more flexible. It's not anticipated that you will be in one job for life, now. So and I think that's quite a healthy way to approach life, in general, not to just limit yourself as such. So.
And how have you found, Susan, your interactions with, how do you work within the AstraZeneca with the smaller startups, with the academics, the trialists, and what are your thoughts on the future of where you might go? Would you spin something out?
Yeah, we do a huge amount of collaboration. So whilst we have a big investment in internal discovery and development, we're also very actively looking always on the business development side about the opportunities we have for collaborations, both in a very early stage. So we have some collaborations. So we have one with a company called Scorpion, which is right in the discovery stage, where they have a technology which is enabling and speeding up the ability to drug targets that have been previously thought to be undruggable. We have late stage business development deals. So a couple of the antibody drug conjugates in our portfolio have come from partnership with a Japanese company, Daiichi Sankyo, which we've brought in. We can bring the global scale of development with the number one pharmaceutical company in Japan, with the number one pharmaceutical company in China. We have a huge global presence. That global capability is what small biotechs will need when they're wanting to turn the very exciting data that you might have at an early stage into something that is accessible to patients across the world. So we do from the very early to the very late and everything in between. We do academic collaborations. We spend a significant amount of money fostering those as well, and some collaborations. So I'll give you another example. We had a drug approved last year called capivasertib, which started off in the very initiation of the collaboration between Aztecs Pharmaceuticals, another Cambridge company that was based on fragment-based drug discovery, and the Institute of Cancer Research in London. And that collaboration, the actual molecule that we discovered came from our own chemistry effort, but the insights into how you might drug that target came from that kind of collaboration. So we do academic collaborations, we do small biotechs, we do large deals at the end, and we're constantly looking. So the idea that we can innovate on our own everything that is needed is just not going to work. So again, we spend a lot of time and effort on the network.
I think networks and collaboration is the only way really to excel now. No one company can do everything on its own. It's just not possible. You see from Susan's talk, the science is going in so many directions. There's emerging areas. You can't be all things to all people. We collaborate widely as well. We have collaborations with pharma companies, with biotech companies, with academic institutes. I think everyone's doing the same. It's the only way you can. I think fundamental premise of what we do these days is collaboration. It has to be.
And is there a particular mindset, personality, set of skills that works well, you think, in a biotech versus maybe an academic setting, or
set of skills in a biotech setting?
Yeah. You have people who come and don't. You're talking to people who may be thinking about their career and where it might take them. What would?
I guess there are different drivers. I suspect there are different drivers in the different industry, biotech and academia. I mean, academia, you've got to be prepared to put in the grants and you're looking for promotions or publications, and maybe that's kind of what you're looking at, and you're not necessarily looking for line of sight to registration of a drug or as such. You're kind of trying to exploit maybe your kind of unique selling points within your lab, your kind of your academic collaborators, and so maybe you want to progress a small part down a natural pathway, but not necessarily take it all the way through to drug approval. So I guess, maybe, I don't know if it's a time versus kind of depth and breadth sort of mentality. I have no idea,
I think there's a lot of overlap at the science level. There's a lot of overlap between all three disciplines. Right? You do your focus. You're doing a PhD. I think the time horizons can be slightly different. I think in academia, definitively when your money runs out. In biotech, you've got a rough idea when your money's run out, but you're never quite sure. And in pharma,
in pharma, you're doing your budgets, planning all the time.
Yeah, exactly, and changing it.
I think we have the budget numbers are different, but the process of the fact that you've got to pitch for the money and the investment that you've got to do, and you've got to make that case very well. It's that is universal. It's just the scale of it. I would just say, in a big company, it's a team sport.
Yeah.
It's a team sport. You've got to be able to go across cultures, across regions, and manage those different things. In a biotech, you have the ability to focus on the smaller set and a smaller team that you can drive and make it. And that's what I think you sort of talk about, the amount of time that you might feel on things that are not immediately productive to you and your specific goals is probably less, so you can have that level of focus. But you've got the scale of the ability to have an impact in a bigger company. But in both settings, they're team sports. And I think, academia, team science is very important as well, but it is possible to be a bit more individualistic.
I think that those, I think there has been a change, and there were a lot of mini empires. I think there was a lot of that kind of insularity sometimes in groups not wanting to share data. But actually, one of the really encouraging things I'm finding in terms of grant awards and it is the network collaborations and now, for instance, experimental cancer medicine center initiatives. Unless your work is actually being translated elsewhere or you're collaborating elsewhere, it's kind of, you have to kind of build those networks and work across sites. And actually that makes a lot of sense. You need that help to recruit different populations, different demographics, if you're doing a kind of a UK trial. And you also need that expertise. It might not all be centred in one place, and they'll bring different intel, with your academic collaborators. It's a bit of a healthier place, I think, from that point of view. That collaboration is actually sought after now. It's being encouraged, which is all good.
Yeah, wonderful. I'm sure there will be some questions from the audience, and we're running a little bit over, but I think we've got 5 or 10 minutes just to carry on. Can I encourage anybody to ask a burning question? Do you want to stand up, just say briefly who you are?
I'm alumna Mishra. I'm actually Ruth's niece, so I'm really delighted.
Oh, wonderful.
And she was a very inspiring woman, and so it's great to see so many women and a man talking today.
They've got a microphone, thank you.
But I wanted to ask about inequality. This was something you referred to. There's many new cancer treatments coming, and they seem to be increasingly expensive. How can we make sure that cancer survival isn't something for the rich, that it's something for everyone?
Yeah, no, I think that's a really important question. So let me just tackle it from the beginning. Again, if you think about how drugs are developed, so quite often we will start off in the initial trial that gets to the initial approval in a patient population that is what we would call later stage. So these are patients who might have had diagnosis, they may have had an initial chemotherapy and surgery. They may have, their cancer's then progressed following that, and they've had 1 or 2 other treatments. And those are the people that are typically going on to the initial trials with the drug. So their outcome and likelihood of outcome is already short. Quite often, the end point that we measure in those clinical trials might be what we call progression-free survival, which is the time it takes until their cancer progresses, okay? For many people in that situation, you're talking about 4 or 6 months. And so what you're looking for is some extension on that as proof of principle. So what you end up with are trials where you've shown an improvement in that of 3 months. So you started with 4 and you've gone to 7. And people will rightly say, well, the cost for that level of improvement is high. But that same drug, when taken into the early stage, can produce 3 to 5 years extension in life, and even the potential for some patients to have a more normal lifespan. So definition of cure is a whole debate on its own. And there are multiple examples of that. The cost effectiveness in that second example that I've given you meets all of the NICE criteria for the cost effective, which, by the way, the NICE criteria for the willingness to pay in the UK has not changed in over 20 years. And if you hadn't noticed, there has been inflation during that time period, right? So I would argue, right, that we are a relatively rich country, and the affordability, the things that we're setting, and we deserve to have, as a community, better outcomes than we're currently pitching for. That would be, my perspective. And again, there's going to be different perspectives on that, and that the value is gained by treating the cancer at the right point in the disease and putting it together with the other. Cure is more cost effective, and so if you can achieve that, it can be still cost effective. So I think what we need to think about is the totality of that infrastructure and the other piece, and not just focus on the one aspect. Now, inclusion and diversity in clinical trials is absolutely fundamentally important. I've said at one point that many of the advances that we've got is through a better understanding of the genomics and aberrations that drive cancer. Many of those genomes are Caucasian that are actually analysed. There's relatively poor representation of the genomes of people from other backgrounds. That's something that we've got to fix. And all of that data and AI that we're going to then apply to that is going to be, you have to make sure that the data set that's fundamentally going into it. So there's many, is representing the patients that you actually want to treat. So there are many different aspects of diversity and inclusion that we need to address. And I think it is fundamentally important. But I do think that it's an achievable goal to have cost-effective cancer treatments that are affordable and available to all. I think that's an achievable goal. Many systems aren't doing that ideally. And I do recognise there's a burden and there's a challenge for that. We have to partner with healthcare systems across the world to sort of make that better than what we've currently got. And, we're working very hard on doing that. We're making some progress, but there's a lot more that we need to do, I think, in that area. So thank you for raising the question, because I think it's a fundamentally important debate that we need to have.
And how do you, Kevin, think about the cost of the
very
drugs you're
challenging.
bringing to market?
I don't really have much more to add.
Because the costs are in the development phase, aren't they?
Oh, it's billions. It's a lot of money to develop a drug. So we have to put the initial investment in. The failure rate is very high. So when you actually, you work out the cost to develop a drug, factoring in all of the failure rate, because you get it wrong more than we get it right, it becomes a very expensive process. But I think we're all trying to figure out how we improve access.
I think I just want to add, though, the question of inequality, even outside of where you're developing drugs, I treat patients in clinic, and I know that I have, for instance, in my liver cancer clinic, I have patients who had a little bit too much alcohol. Perhaps they say, they've had cirrhosis as a result of it, or they've had quite, they might have had viruses in the past. And there's a very wide spectrum of patients who present with this kind of cancer in very, very wide socioeconomic kind of disparity. There's some people who have had drug addiction and hepatitis viruses, and then some people who have actually had a pretty comfortable life and maybe overindulge in certain things, and it's possibly the kind of certain stigmas associated now with this liver cancer side. Exactly. But in that, I might have a drug I can give to a patient, and I might say, you need to come in, get your blood test done, get your treatments, and I can see you, etc. And they might say to me, well, doctor, you can tell me I need this, but you can tell me, but I don't have £10 and I don't have 10p to come in. I just can't afford to come in. It is that level of inequality that they can't even access the drugs which we know will work for them sometimes. It's a lot to work for, a lot of work going in there, even before, outside of the stage of the research and the development, that the inequality is rife, and how do we address that in society? That's a totally bigger picture, but I see it I see it frequently in front of me, and I think it's very important that actually the trials, and I find it very easy for, working with a lot of pharma companies where they say, well, if you have a patient from a certain demographic where they would not have access to these new drugs, we want to make sure they have access. So we'll cover this. We'll cover their cat care. We'll cover their hotel stay if they need to, so that they can participate and have access to these drugs. It's very important. It's not so easy for academic trials because a lot of these are running on tuppence halfpenny. We're counting every penny for the research side, and we can't cover that. We know that we're not going to be able to recruit some patients because literally they can't afford it. Yeah.
This is why some of the digital tools, I think, can be helpful, because it means that you can reduce the number of visits that need to come. And I also think that it means that we've got to actually make clinical trials available at more community-based centres. So you can often have a network. Yale network is one example. For example, so New Haven, whilst Yale University is situated there, New Haven actually has a high degree of deprivation within that city, but they also have a network across the state of Connecticut where they can connect out with the clinical trials. That kind of system enables you to get access to people who cannot afford the transport to go to the clinical trial centre, and that's one way in which you can improve it. The digital tools which you can give people access to means that they can document many things. Blood tests can be done closer to home. You don't actually have to have the scan necessarily at the central hospital. There are ways in which you can make clinical trials more accessible, and I think we have to think about many aspects of that.
Lots of aspects that are coming into the diversity and quality. I think probably one more question, so otherwise food for those who are going on to formal hall, and that I think we're staying on, so there might be opportunity. Another question? Is there another question?
There was one more.
Yes, please do. Yeah. Yes. Thank you. And again, if you could just introduce yourself.
Lucy, and I work here at the college as head of communications. I just wanted to ask you, in the light of the King being diagnosed with cancer, what do you think the impact of that will be on awareness, conversation around cancer in this country?
Well, to start with that, cancer it's so common. It's so common, and actually, if the King can get it, and talk about it, well, I think he's done a very brave thing, because a lot of people do want to hide away from their cancer diagnosis, and they want to squirrel themselves away at home or, just in the hospital and just get through it, one mind. But actually, for someone like him to say this is something he's found, he's getting on and treating it, and, maybe it would make you think, well, if he can get it, and he's had a very privileged lifestyle, he's done everything probably right, and it can still affect someone like him. I think that this is, it's very sad for him, but he's doing a great thing by kind of publicising it and making it, I think it's going to increase maybe screening, maybe increase, calls to the GP perhaps, or approaches, because people might say, well, maybe I do need to think about it, because once, someone who's quite in the public domain has a cancer diagnosis, it does raise public awareness. It does kind of suddenly broach your awareness, because a lot of people don't want to think about it. They really don't want to think it's going to affect them, but the chances are it's 1 in 2 pretty much now. So
1 in 2. Well, on that sobering thought, it's very good that we're developing more treatments for it. And so I'd like to thank, maybe you could join us in thanking Susan and Kevin and Bristi for joining us this evening to talk about how we tackle cancer.
Thank you.